For this session, we're going to talk about enzymes. Although we have talked about enzymes a little during our past lecture, we have not really delved much into the details of this important biomolecule. So as we said before, enzymes are also proteins, okay, and they are organic catalysts, okay, meaning they facilitate a particular type of chemical reaction without being affected in the process. So that means that we can still obtain the enzyme after the process, after the chemical reaction. Enzymes. So number one, enzymes are proteins. Enzymes follow physical and chemical reactions of proteins. And they are heat labeled, so that means they are destroyed by heat because proteins, as we remember, are denatured by heat. So therefore, enzymes being protein are also destroyed by heat. Number three, they are water soluble. Number four, they are precipitated by protein precipitating agents. So that means that this is also a form of denaturation. So by adding certain acids or um, alkaline solutions or even salts, it can lead to precipitation of enzymes. So that is still denaturation. And they are about 16% nitrogen. Now, enzymes, if you remember, the names of enzymes that we have introduced last time are, most of them end in ASE. So that, for example, lactase, amylose, amylase. So for enzymes, okay, they are given this kind of name, but it is rather uh, uh, it doesn't have any basis, so it is ambiguous. It's not really clear how enzymes are named. So therefore, a particular group of biochemists came up with certain rules to follow in naming an enzyme. So it is a digit system. Digit system. It's made up of 4 digits separated by dots. Okay. So it starts with EC, capital E and C, which means enzyme class, then followed by 4 digits. So the first digit represents the class, the second digit represents the subclass, the third digit is the subgroup, and the fourth digit is the number of the enzyme K in the list. So sometimes you may encounter several enzymes having the same characteristics. So it's because of the existence of what we call isoenzymes. So they are isomers of enzymes. So that means they are the same enzyme, but they differ. They differ in, for example, the source where they are found, okay, or particular organ where they are produced. The classification of enzymes, the different classes of enzymes. The first one, or class one, is oxidoreductase. We can more or less deduce the kind of chemical reaction it carries out. So oxidation and reduction, so redox reaction, right? So the chemical reaction is represented this way: AH2 plus B forms A plus BH2. Is more like a replacement reaction. So a specific example, we have um, alcohol plus NAD, okay, oxidized NAD forms a aldehyde. Aldehyde plus NADH, so this is reduced NAD plus hydrogen, free hydrogen. So the enzyme that is used to convert alcohol to aldehyde is called alcohol dehydrogenase. Okay, its IUB name is alcohol NAD oxidoreductase, and the code number is EC1.1.1.1. So makikita natin. So of course, you are not really required to remember all of them. So just maybe some examples. Like in this case, alcohol dehydrogenase, the very first enzyme in the listed in the IUB, EC 1.1.1, oxidoreductase, alcohol dehydrogenase. So oxidoreductase may also oxidize substrate by adding oxygen. Okay, for example, we have oxidases, oxygenases, and dehydrogenase. Second type of enzymes are the transferases. So it's obvious what they do. They transfer a particular functional group to another place, to another part of the substrate. So this can be from one substrate to another substrate. And this can be represented by this chemical reaction. AR plus B forms or yields A plus BR. So that means that the R was transferred from A to B. Right, so a specific example, hexose plus ATP forms hexose 6-phosphate. So that means that one of the phosphate from ATP was transferred to hexose, forming hexose 6-phosphate plus adenosine diphosphate. So the enzyme that is used to do this kind of reaction is called hexokinase. Okay, and the name of this enzyme, okay, is also ATP hexose 6 phosphate transferase. Next, hydrolases. Hydrolyze. So the word hydrolase indicates that this kind of enzyme performs hydrolysis. So they can hydrolyze esters, ethers, peptides, or glycosidic bonds by adding water, okay, and then breaking the bond. Okay. So an example here would be acetylcholine plus water forms acetate and choline. So that means that acetylcholine is divided into forms, two forms, so we can say that this is a kind of um, decomposition reaction. The enzyme is acetylcholinesterase. Maybe you've heard of this, acetylcholinesterase. So this is found in acetylcholine is found in skeletal muscles, and acetylcholinesterase is the one that is responsible for uh, encoding or decoding the information from acetylcholine. So you remember this acetylcholine is um, the substance responsible for muscular contraction. So and then systematically we can also call this acetylcholine hydrolase. So what else? All digestive enzymes are hydrolases. So try to recall what are the different digestive enzymes. So enzymes that are produced in the gastrointestinal tract, including the enzymes that are produced by the pancreas. So remember the enzymes responsible for protein digestion, trypsin and pepsin, the peptidases, the lipase, amylase. So those enzymes are all in terms of classification hydrolases. Next, number 4, bases. So these enzymes can remove group from one substrate or break bonds by mechanisms other than hydrolysis. So the action performed by this is also breaking bonds. For example, fructose 1,6-biphosphate is converted into glyceraldehyde 3-phosphate plus dihydroxyacetone phosphate. Mention the enzyme here, but anyway, so that is an action performed by a lyase. We will have more examples later on. Isomerase, isomer from the word isomer. So they can perform a change in the substrate, okay, by For example, optical, so the L and the D form, geometric conversion. Okay, so that can be formation of the cyclic bonds, positional isomers, so changing the position of the functional groups or substrate, but still the number of atoms remain the same. So examples of this would be the racemase, epimerase, the cis-trans isomerase. So example of a chemical reaction undergone by the by isomerase is glyceraldehyde triphosphate is converted into dihydroxyacetone phosphate. So the enzyme here is triose phosphate isomerase. So we know that glyceraldehyde and dihydroxyacetones are isomers. They are isomers. They are both triose with the presence of the phosphate group in this case. As classification, they are the ligases. So ligate. So maybe you've heard of ligation. So enzymes that tie or link two substrates together, okay, they are ligases with the simultaneous hydrolysis of ATP. So in this case, it's required for the utilization of adenosine triphosphate. Acetyl coenzyme A plus carbon dioxide plus ATP forms malonyl coenzyme A. This is the product. And ADP plus inorganic phosphorus. The enzyme that was used here is acetyl coenzyme A carboxylase. Right, so as we have mentioned, there are 6 types of enzymes: the oxidoreductase, transferases, hydrolases, lyases, isomerase, and ligase. So these are the actions performed by the oxidoreductase. They transfer hydrogen or add oxygen. Okay, so these are the examples: LDH, lactate dehydrogenase or LD, uh, glucose 6-phosphate dehydrogenase. So notice the word dehydrogenase and the oxygenases or the oxidases. They are all oxidoreductase. Transferase. So in the name itself, you can see the word transfer. For example, aminotransferase. Let's say aspartate aminotransferase. So that means it is transferring an amino group. Okay, aspartate aminotransferase. Alanine aminotransferase. So those are examples. The kinase or the kinase. Okay, what else? Hexokinase. So as we mentioned earlier. And then hydrolase, as we said, they cleave the bond and add water in the molecule. Okay. One example is acetylcholinesterase, and then trypsin is also another example. And as we have mentioned, all of the enzymes that are found in the gastrointestinal tract. Lyase. So this cleave, okay, the molecule without adding water. So union difference number 3 and number 4. Okay, for number 3, water is required. For number 4, it's not required. So we have aldolase, HMG coenzyme A lyase, ATP citrate lyase. Okay, so hydratase. Yeah, so those are examples of lyases. Number 5, isomerase. So the racemases and the epimerases example, triphosphate isomerase. This was the given example earlier. And then for ligases, ATP dependent. So that means it's required to utilize ATP. Okay, an example that we had was acetylcoenzyme A carboxylase, and additionally we have glutamine synthetase and PRPP synthetase. So those are the examples of enzymes that belong to each type. Okay, class 1 to class 6. So we did not really mention of the specific IUB names for each, so the digital name. However, in clinical chemistry, you will encounter enzymes again, and you may need to remember or memorize the code for those biologically important or medically important enzymes. All right, so just to just to, you might get confused because of the name, synthetase and synthase. So synthetase are ATP dependent. So if they are dependent on ATP, so they must fall to class 6. So, and then examples of this would be carbamoyl phosphate synthetase, arginino succinate synthetase, PRPP synthetase, glutamine synthetase. So you can see in the name the word synthetase. What about synthase? Synthase are enzymes that can catalyze biosynthetic reactions. So synthesis reaction, similar to synthetase, except that there is no requirement for ATP. And in terms of classification, they belong to the ligases. Other than ligases, they may fall into other classification except for the ligases, other than class 6. So examples are glycogen synthase, alanine synthase, so that means they can produce these substances in the absence of ATP. So, okay. An enzyme is made up of not just the protein molecule. Sometimes it is, not just sometimes, but all the time, it requires other parts. So a complete enzyme is called a holoenzyme. A holoenzyme. Sometimes this holoenzyme may need a metallic coenzyme or an organic coenzyme. So, alam natin yung pagkakaiba. Organic, ibig sabihin na carbon-containing substance. Pag metallic, of course, metal yan. Right. So, the protein part of the enzyme is called the apoenzyme. The apoenzyme. Okay, what else? So, it has two parts. The prosthetic part is the coenzyme. So this is the heat stable part, and the apoenzyme, or which is the protein part, is heat label. So that means it is destroyed by heat. Okay, we know that. Okay. Okay. Coenzymes may be divided into two groups. Those taking part in the catalysis by oxidoreductase. Okay, by donating or accepting hydrogens or electrons. And the second groups would be those that are important in transferring groups other than hydrogen. Okay. So, salient features of coenzymes. So we have 7 important features of coenzymes. So the protein part, as we know, this is the apoenzyme, gives the necessary three-dimensional infrastructure for chemical reaction. So we remember that there are two theories. Actually, there are three theories, but we mention only of the two important theories for enzymatic reaction. Okay, the first one being um, uh, wait. So number two, coenzyme is essential for biological activity of the enzyme. So without it, it might not be able to perform the reaction needed by that can be performed by the enzyme. Number three, it is low molecular weight organic substance. So as I told you, this is an organic substance and it is heat stable. Later on, we will give some examples of coenzymes. Generally, coenzymes combine loosely with the enzyme molecule. So that means later on, it can separate with the enzyme, okay, as it carries out the enzymatic reaction. Number 5, inside the body, the coenzyme is released by the apoenzyme and bind to another molecule. Okay. And number 6, one molecule of coenzyme is able to convert a large amount of substrate molecules with the help of the enzyme. Number 7, most of the coenzymes are derived from vitamin B complex. So this is one importance of the vitamins. They act as um, coenzymes. We have here the first group of coenzymes. So again, they are organic. They are organic substances, right? So what do you have here? Coenzymes may be considered co-substrates or secondary substrates. Okay, and for example, lactate. Okay, lactate, which is a product of anaerobic respiration, can be acted upon by an enzyme lactate dehydrogenase. Okay, in the presence of a coenzyme, and it is going to be reduced. So that is an example. So we have some specific examples of these coenzymes. NADP, NADPH. So yung may H is reduced. Yung wala, that is oxidized. FAD, FADH2, FMN, FMNH2. So yan yung mga examples ng ating coenzymes. Coenzymes. So they are nicotinamide adenine dinucleotide phosphate. So ganun ang ganun ang name. Okay. Nicotinamide adenine dinucleotide. So this is a coenzyme from nicotinamides, which is a member of vitamin B. Right, so this is the structure. Okay, we don't really have the structure for this enzyme, but this is an example of a process that can be performed by lactate dehydrogenase in the presence of NAD. So lactate can be converted to pyruvate, and pyruvate can be converted back to lactate. Glyceraldehyde can be converted into biphosphoglycerate. So that is another reaction. So we can say that it is a reversible reaction. Okay, and 2 hydrogen atoms are removed from lactate. Okay, 1 hydrogen and 2 electrons are accepted by the coenzymes. So the hydrogen is accepted by the nicotinamide group. Okay, so as we said, there are two ways by which a chemical reaction and enzymatic reaction can happen. One is the transfer of hydrogen, and the other one would be the transfer of other groups. Okay, for example, other than hydrogen. So the other functional groups may be transferred also. Okay, so still the enzymes that are needed here belong to vitamin B complex group. So adenosine triphosphate. So we already know ATP is a molecule. It is a molecule. It's not really a coenzyme, but it is a molecule that is necessary for the transfer of energy or production of energy. Next, we have the cofactors. So cofactors are also coenzymes, okay? Except that they are usually metallic ions. Okay, so pag coenzyme, organic. Kapag cofactor, metallic. So, let's see. Copper is needed in tyrosinase. So copper. So copper is a metalloenzyme. So if it is bound to, okay, if it is bound to that enzyme. So calcium can also be important. So calcium is necessary for the activation of pancreatic lipase. So also magnesium, chloride. So those are examples of ions that are necessary for the activity of the enzyme. We have here some examples of metalloenzymes: zinc, magnesium, manganese, copper, iron, calcium, and these are the enzymes that are that require the presence of these and metals. the last slide. Okay, although I did not include that there are certain factors that can affect an enzyme reaction, maybe we can talk about that in the laboratory. Okay, thank you for um, watching, listening to this video. Goodbye.